Assalamualaikum and good day to everyone. So today I'm going to continue on another subtopic in enzyme preparation, which is on cell breakage. So other terms for cell breakage is cell disruption or cell lysis. All right. So we have covered a little bit on solid liquid separation, which is part of downstream processing of enzyme preparation. So after we separate the biomass from the liquid portion, then we will look at whether the enzyme of interest is extracellular or intracellular, in which case for intracellular enzyme we need to perform the cell breakage in order to release the enzymes of interest from the cells and further purify it. So the energy needed for the cell breakage vary. Some cells are sensitive to gentle treatment, for example the osmotic shock. So what are the examples? Some gram positives, bacteria, as well as some animal cells as well. So other cells are more resistant. So what are the example of the resistant cells? There are the gram-negative cells, bacteria, as well as the yeast and the fungi. Sorry, my daughter wants to come.
Ada Afia lah.
Okay, Mami nak buat kerja. Afia duduk sana nah. Alright, sorry for the disruption. Okay, so let's continue with our slide here. So some cells are sensitive to gentle treatment using osmotic shock, but the other cells are more resistant, so we need a more energy invasive method, and they are very violent in terms of the method to disrupt the cells. So if you look at this table here, there are mechanical and non-mechanical types of cell lysis. Okay, so because for microbial cells, the mechanical and for enzymes, the mechanical methods are usually used in order to perform large-scale lysis. So we will only cover the mechanical method for our in our subject. So we will look at this part here, which is on the sonication, bead mill, and high pressure homogenizer. Okay, so the non-mechanical method includes physical method, chemical method, and biological method. So one of them is the osmotic shock, which I've just mentioned to you just now, and heating as well as cavitation. For chemical method, we use detergents and alkali, and for biological method, we use enzyme, for example, lysozyme. Okay, so some solution, lysis solution, even though we use the mechanical type of lysis, can also combine or add in a little bit of the non-mechanical chemicals to the lysis solution before they were subjected to the mechanical method. Okay, but if you talk about large scale, if the chemicals are expensive and we can do away without it, then they won't put it inside. All right. So there are many types of cell disruption techniques available. Some are suitable for small scale, some can be upscaled to large scale. So we will look at that in much more detail. So it's very important to choose a suitable cell disruption strategies to avoid damaging the enzyme. So the cell disruption methods, there are many types of cell disruption method. So again, the choice of cell disruption method depend on the susceptibility of the cells to disruption. So you need to understand the differences between the gram-negative bacteria, gram-positive bacteria, yeast cell, plant cell, animal cell, in order to choose the most suitable method to disrupt the cells. And you need to also know the product stability, whether they can withstand heat that are generated when we disrupt the cells, and whether they are thermostable. And also when we have disruption of the cells, we will produce some cell debris here. So we need to extract the enzymes from the cell debris. So we need to know the speed of and the ease of extracting the enzyme from the cell debris. And the speed and cost also will be affecting our final choice. So the ideal large-scale disruptor will be able to do all these things that is listed here. So we want ideally for the large-scale cell disruptor to be able to disrupt tough organisms, and the mechanism of how they work should be well understood for troubleshooting. And of course we want it to be easily sterilizable so that we don't have contamination, and it needs to be containable and validatable. So if we include some control measures, QA, QC, it is validatable, and it is easy or amenable to being made automatic for the ease of process. It's continuous and compact, and it also needs to be economical. Okay, most of it cannot be any cell disruption method are not perfect, but hopefully it can have most of the points here. It can satisfy most of the points here. Yeah. Okay. So let's see the breaking cells and tissues. So this is a figure that showed four types of not mechanical, four types of okay. So these are examples of the four commonly used procedures to breaking cells and tissues. So it's a nice figure, so that's why I like to show it to you here. So when you have a cell suspension or tissue, so you can use this four commonly used procedures. So the first one is you use sonication, where you break the cells with high frequency sound. And then the second procedure commonly used is using a mild detergent. So the detergent, because it can interact with the uh, lipid membrane, then the detergent can make holes in the plasma membrane. Okay, and then the third one is to use a pressure to force cells to go through a small hole. So this one we will cover as well. So we call this the French press, or the hold on, let me see what is it. Forgot already. The high pressure homogenizer. Okay, it's a high pressure homogenizer. All right, so that's the three commonly used procedure. And the fourth one is to shear the cells between a close fitting rotating plunger and the thick walls of a glass vessel. So this is a type of shearing method in order to shear the cells. So what we have here, we usually include some beads, beads, glass beads, inside the chamber, so that you can have shearing of the cells even better. Okay, so we will cover that as well. So when carefully conducted, then you can have the membrane enclosed organelles intact, but the cells are disrupted. Okay, so the resulting thick soup here is called a homogenate or an extract, and it contains large and small molecules from the cytosol, such as enzymes, ribosomes, and metabolites. Okay, so I think it's quite a long video already, so let's stop for a while and I'll continue later.